story highlights. In the 1990s, Lee Sweeney made headlines with his Schwarzenegger. Soon received calls from multiple athletes in hope of a genetic performance boost. CNN. In 2008, an Olympic year, Lee Sweeney's phone was ringing non-stop. For a busy physiologist at the University of Pennsylvania's Perelman School of Medicine, that may be expected, but the reason behind the calls wasn't exactly run-of-the-mill. The people on the other end of the line were athletes in search of a particular kind of fix. They wanted him to dope them via their genes. In the late 1990s, Sweeney made headlines because of his research on Schwarzenegger mice, which were up to 30% stronger than their average counterparts. Sweeney had been able to isolate the gene responsible for activating a protein, IGF-1, that controls muscle growth and repair. The main focus of his experiments was on how to limit the deterioration of muscles with age, but the results also appealed to athletes in search of a performance boost. Read more. Word got out, however, that he was not interested. Bodybuilding drugs sold online often contain unapproved substances, study says. Ahead of this year's Commonwealth Games, which started April 4th, Sweeney's was not such a hot number for athletes in search of an unfair advantage, possibly because he is now an advisor for the World Anti-Doping Agency. At the beginning, when we first started publishing on this, we did get contacted by high-level athletes, said Sweeney, who's also director of the University of Florida's Myology Institute. These days, it's mostly bodybuilders and people desperate to increase their performance or abilities. Back then, gene therapy, defined as a technique of using and manipulating genes in order to treat or prevent diseases, wasn't as established as it is today and wasn't recognized as enough of a threat to be listed as a banned practice in sport. Responding promptly to the possibility, in 2002, the Anti-Doping Agency established gene and cell doping panels of expert scientists to discuss how best to head off the problem, but it soon became known that gene therapies could one day be used for much more than disease. In 2003, the organization banned gene doping, which it defined as the non-therapeutic use of cells, genes, genetic elements, or modulation of gene expression having the capacity to enhance performance in sports. Who's really old? This new frontier of doping presented a simple and dark idea. What if there was a way for dopers to never be caught? Now, almost 20 years later, the technology has finally been used to treat patients with rare diseases such as severe combined immunodeficiency, chronic granulomatous disorder, hemophilia, blindness, cancer, and neurodegenerative diseases by transferring missing genes into skeletal muscles, Sweeney said. It could be done today in athletes if some company and government would put the resources in to make it happen, he said. So because of that, it is now at a point where potentially it could be used by athletes. Getting inside your genes. In the case of the Schwarzenegger mice, Sweeney used the classic method of gene therapy, in which he modified the animal's DNA using a virus to deliver and insert the required gene that would make the mice stronger. Genes are delivered into an organism using a vector, the most common being viruses, like that used by Sweeney, which have been modified to be safe and no longer cause disease. The vectors carry the desired gene into targeted cells and there unload the genetic material, which in turn instructs the organism to produce the protein the gene encodes. One example of a protein well known to athletes is erythropoietin, commonly known as EPO, which regulates the production of red blood cells in the body, increasing hemoglobin and oxygen delivery to tissues. With the injection of external EPO, elite athletes, often cyclists, have been enhancing performance for years, but authorities have caught on. A doping urine test kit is seen on February 25, 2015 in Châtenay, France. Anti-doping controls can now detect external EPO efficiently through blood and urine tests. If extra EPO is being produced organically by a cell's machinery, however, it is almost impossible to detect as a banned substance. But the technology is not quite at that level yet. Making the viruses that are involved in doing the gene transfer is still difficult, Sweeney said, highlighting that the science is still complicated and not something athletes could readily do at home. What the NFL could learn about drug testing from the UFC. Another way to dope an athlete's genes is through CRISPR or CRISPR-Cas9, a technique that allows geneticists to edit specific parts of a person's genome by removing or altering sections of DNA, also known as gene editing. The technique is rapidly developing, leading to a World Anti-Doping Agency announcement in October that it was expanding its gene doping ban to gene editing agents designed to alter genome sequences and/or the transcriptional or epigenetic regulation of gene expression. The ban went into effect in January. There's a couple of ways you can use CRISPR-Cas9. One is to take cells from a person, modify those cells and put them back into the person, and that is probably the safest way to use it, Sweeney said. The other way to use it, which is to modify your existing DNA in the body, is potentially very unsafe. False kit of genetics editing with CRISPR-Cas9 technology. Sweeney pointed out that scientists do not know what unintended consequences could come from changing a specific gene in an individual, meaning the technology is not even ready for trials in patients with lethal diseases. In the case of gene doping through gene therapies using vectors for delivery, it's relatively easy to look for an extra copy of a gene and confirm that an athlete has been doped when you have access to a biological sample, such as blood, said Olivier Robin, Senior Executive Director of Sciences and International Partnerships at the Anti-Doping Agency. In particular, Robin said the agency looks at white blood cells and has developed a methodology that can be applied to search for different genes. Americans wary of gene editing, brain chips. Synthetic blood gene editing is a little more complex than gene therapy, Robin added. Further detail was not provided as it is kept confidential in order to catch athletes, he added. The anti-doping agency is working on strategies to reveal the possibility of people editing their genes for performance enhancement, he said. Robin highlighted that most of the agency's efforts focus on white blood cells as pretty good markers of gene manipulation because some evidence of manipulation will usually end up in the blood. Asked what it is doing to monitor and test athletes for gene doping, the International Olympic Committee did not comment directly but said, "We have nothing to add to the section on gene doping in what is prohibited list. The question now is whether the first few cases have, in fact, happened without our knowledge. Modern occurrence. I never heard anything about it except for one time, and it was around 5 years ago," said Sebastian Weber, coach of four-time Union Cyclist Internationale world champion time trialing Tony Morton. There was some buzz around a substance called ICAR, or 5-aminoimidazole 4-carboxamide ribonucleotide. Rafael Nadal, Spaniard, happy to make his drug tests made public. ICAR is a performance-enhancing drug that the French anti-doping agency suspected was used in the 2009 Tour de France. It stimulates mitochondria, the part in the muscles responsible for aerobic energy production. In cycling, for gene doping to be effective, techniques should target both EPO levels and red blood cell production to have a higher oxygen delivery to the muscles, Weber says, but they would also need to increase the mass and number of mitochondria in order to actually produce energy from that oxygen. Just because you have more oxygen, it doesn't necessarily mean you also have the capacity to produce energy out of it, Weber said. As ICAR was a drug, it wasn't gene doping, but it led people to wonder about what was next, he says, after this first step towards stimulating the body's mitochondria. That was the only time I heard people talking about the possibility of gene doping. Tailoring to your genes. There are other ways. Genetics, and a deep knowledge of them, could help athletes improve their performance by understanding their physiology. For example, Project Genesis focused on how applied genomics in elite sports can improve performance, and its offshoot, the Rugby Gene Project, are trying to identify which genetic characteristics help athletes succeed. British doctor denies report that he doped 150 sports stars. We recognize it is not only genetic, said Drive. Alan Williams, an exercise geneticist at Manchester Metropolitan University in the UK who works on both projects, training, diet and other lifestyle habits are massive factors. But along with that, the evidence is that it's impossible to have success in sports without some genetic factors in your favor. The researchers of these projects are hoping to identify which genes help or hinder athletes in their specific disciplines to develop their skills in a more tailored way. For example, if an athlete has shown to have a higher genetic vulnerability to tendon injuries, scientists and coaches could reduce certain aspects of their training load over the course of the season, give longer rest periods, reduce the number of matches played in a season, or provide specific exercises and prehabilitation workouts. But Williams points out that the field is still at an early stage. This picture where certain genes, or even 2 or 3 genes, are related to a particular characteristic like the tendon injury is still a small bit of a bigger picture, he said, so it's very important that this information that is available is put into context. At different point of view, some experts argue that we're looking at it all wrong and that athletes will always use the most modern technology to seek out an advantage, illegal or not. Follow CNN Health on Facebook and Twitter. See the latest news and share your comments with CNN Health on Facebook and Twitter. Modern sports have been principally valued on the basis of record-breaking and being able to witness extraordinary performances, said bioethicist Andy Maya, the University of Staffordshire in Science Communication and Future Media and author of Genetically Modified Athletes, Biomedical Ethics. Gene doping and sport. Even if it's not a world record, it's about trying to see something special in what humans can do, and often that is about transcending boundaries. We give athletes all sort of technology to do that, added Maya, who readily claims to disagree with anti-doping. Instead of the current scenario in which anti-doping keeps trying to catch up with doping, Maya suggests a safer form of performance enhancement. If we can have a system where enhancement was actually medically supervised, then I think that is a much more safe and healthy. Nick Buska is a triathlon coach, personal trainer and CNN Sport and Health contributor.